Good morning to all of you. So, yesterday we did semi-conservative mode of replication in DNA. The experiment by Messelson and Stahel, and as I told you that Messelson and Stahel, they wanted to prove that the replication of DNA is semi-conservative. So in the year 1958, these two gentlemen, Messelson and Stahel, they wanted to prove that replication of DNA is semi-conservative. By semi-conservative, what do we mean? Semi-conservative means out of the two strands of DNA which are synthesized, one strand is old and one strand is formed new. So one strand is, or the template strand, one strand which is the template strand, this is old and another strand is formed new. This is what they wanted to find out. This is what they wanted to prove. So this is the experimental proof of semi-conservative mode of replication of DNA. Now, what did they use here? They used here, as I have already told you, what did they use? They use heavy isotope of nitrogen. That means the normal isotope is N14, but what did they use? In place of this, they used N15, which is the heavy isotope of nitrogen in the solution NH4Cl. NH4Cl, and this had this heavy isotope of nitrogen. And this can be, isolated, or this can be identified on the basis of density gradient centrifugation. Density gradient centrifugation. So centrifugation, as I have already told you, this is taken in a test tube and rotated at a very high speed. When the substance which is of high density, it settles at the bottom, and the substances of low density, they remain on the upper surface or upper side of the test tube in the centrifuge. Which test tube is used? That is special test tube, which is known as centrifugation, centrifuge tube. And the centrifugation was done in cesium chloride, which is CsCl, cesium chloride. The centrifugation was done with cesium chloride. Now the bacteria which was taken here, it was E. coli. The bacteria which was used, it was E. coli, a very common bacteria, Escherichia coli. This bacteria, Escherichia coli, it was allowed to grow. I think you are not able to see little of it. Let me adjust it first. You're not able to see the top of it. Yes, now it is clear to you all, I think. Now it is visible properly. So the bacteria which was used for this process, for this experiment, it was E. coli, and the bacteria E. coli, it was cultured in a medium. First of all, this is taken, and then this E. coli, it was cultured in a medium containing NH4Cl, where this N was the heavy isotope of nitrogen. So it was cultured in a medium containing NH4Cl, and it was allowed to grow for many generations. Allowed to grow for many generations. Why it was allowed to grow for many generations? It was allowed to grow for many generations so that both the strands of DNA of E. coli gets labeled with this N15. It was allowed to grow for many generations so that both the strands, they get this N15. That means this N15, it was allowed to grow for many generations so that N15 from NaCl gets incorporated in both DNA strands, so that the N15 gets incorporated in both DNA strands. Because what will happen? If this is allowed to grow in NH4Cl, replication will take place, and when replication will take place, because there is in this medium NH4Cl is there, and NH4Cl which particular N15. So when the replication will take place, one time will come for many generations when it will be allowed to replicate in this medium. One time will come when both the strands, this strand and this strand, both the strands will be in these strands, N15 will get incorporated. We get N15 in them. So this was done color. So because this was allowed to grow for many generations, still both the strands get N15 incorporated into it. So finally, what type of DNA we got? We got a DNA which I'm using the red color to show that the DNA N15 has been incorporated. So if you see, I have used the red color. N15 has got incorporated. So what do we have here? We have in these strands N15, the heavy isotope of nitrogen. So the E. coli was allowed to grow for many generations in a medium containing NH4Cl with nitrogen as the heavy isotope N15, and these were allowed to grow for many generations till both the strands of their DNA gets N15 in them. They are growing in medium of N15. So what will happen? The DNA will get incorporated into the N15 will get incorporated into their DNA. And because the replication will keep taking place, strands of DNA of E. coli will have N15 in them. E. coli got N15 in them. The N15 has been incorporated into them. Now what happened? When this was centrifuged, in the centrifuge tube, this is a centrifuge tube, this heavy isotope of nitrogen, which is present now in both the strands, which place did it take? When it was centrifuged with cesium chloride, and what was used with this cesium chloride? CSL. When it was centrifuged using cesium chloride solution, this N15, it got settled somewhere here. The DNA strand got settled down here. at a lower level, because this settlement is on the basis of density gradient. So N15 is a more denser isotope of nitrogen. So what happened that it got settled here. So if you have your NCERT book, on page number 105, you will find the heavy isotope of nitrogen. This is the heavy isotope, and it got settled here in the centrifuge tube. Okay, because both the strands, they have N15. N15, both the strands, they have N15 incorporated in it, and this N15 is the heavier nitrogen, heavy isotope of nitrogen. It was centrifuged according to density. It took a lower place in the centrifuge tube. It has taken a lower place in the centrifuge tube. Now what happened? Now this particular this thing which we got, that means both the strands of DNA labeled with N15, this was then allowed to grow. This was transferred and in a medium with normal NH4Cl. So this is transferred. Now this was step 1. This was stage 1. This is stage 1 when it was grown in for many generation in N15 medium, and both the strands got N15 incorporated in them, then centrifuge. So it took this place in the centrifuge tube. Now in the stage 2, what was done in stage 2, these E. coli, which has both the strands of this, both the strands having N15, this E. coli which has both the strands with N15, this was allowed to grow in normal nitrogen isotope. That means N14. This is normal H4Cl. In the normal medium, this was allowed to grow. First, it was allowed to grow in the heavy isotope in an N15 H4Cl. Then after this is the same. When both the strands got N15 in them, they were allowed to grow in normal nitrogen solution, which is NH4Cl, where N is the normal isotope, which is N14. When it was allowed to grow there, what happened? Replication took place. When suppose I mark the N14 with black color, and to show you what happened loud in a normal medium, then what happened. See, when it was allowed to grow in the normal medium containing N14, in that case, what happened that this replication took place, and when replication will take place, this red one, of course, this red strand, it will open up. Both the strands will open up. Second strand will also open up. So one strand is old. That means one strand has N15 in it. This is N15. Out of these two, both have separated for replication. Okay, these both were N15. N15, N15. The red color signifies N15. Now, when it was allowed to grow in N14, the stage 2, when it was allowed to grow in N14, now it is allowed to grow in N14. So the new strand which is synthesized, it was N14 type. It had N14 incorporated into it. That means one strand was old strand which has N15, and the newly synthesized strand which was grown in the normal solution of NH4Cl, it has N14. So one was old having N15, then it was grown in NH4Cl having N14, so new strand will have N14. So finally, what did they get? They got strands like this. See these two strands they have got? They got strands like this. One was red, one was black, and another strand, because black is N14. Now they have, and one was old. One strand was old. The red color. So same opened up. This is the old strand was N14, and it was this in color, black in color. So black one is the new strand formed, and the red one is the old strand which I have made. So after application in the normal medium, this happened. Is it clear to you? Is it clear to you? These strands which are formed are known as hybrids. These strands which are formed are known as hybrids, because one strand is the N15 type, another strand is N14 type, one strand is N15 type, another strand is N14 type. Is it clear, children, till here? Till here, is it clear? Till the formation of hybrid, is it clear? Yes, I want your response. Okay, Sukhsahaj is saying yes, ma'am. Yes, more response. Is it clear? I'm not getting your response. You want me to repeat it? Okay, clear. Fine. So these hybrids were formed. Now, when the centrifugation of this was done, this is stage 2. This is stage 1. When the centrifugation of stage 2 was done. So here I carry on with centrifugation. Centrifugation at stage 2. When stage 2 centrifugation was done. Stage 2. And in stage 2, what have we got? We have got hybrids. Hybrids means one old N15 types. This one is N15. The red one is N15. It has got N15 incorporated. The red 14 incorporated. So these are hybrids because one strand has got old N15 and the new strand has got N14 because the solutions were changed. Before it was N15 solution in which it was cultured, and then it was N14 medium in which this was cultured. So now when centrifugation was done, after centrifugation in the centrifuge tube in stage 2, if this is centrifuge tube, the denser one or the heavier one will take the bottom position, but this is not completely heavier. This is an intermediate type. This is one strand heavier, another strand light, one strand heavier, another strand light. So which position will it take, above this or below this? It will take above this. So that can be somewhere above it. That means that can be somewhere here, an intermediate type, which half I'll draw with black color and half I'll draw with red color. So this, it was up here because it was not as denser as N14 type. So this was received, and this was a hybrid having N14 and N15, N14 and N15 strands both. So this was a hybrid, and in the centrifuge tube, this was the position which was taken by this. This was the position which was taken by this. This is the stand. Here also I haven't made the stand. And this is only N15. So it took a lower position because it was denser. This is the density gradient centrifugation. Then after that, what extracted after the transfer? Then it was again the N14. Again, what was taken was N15, N14 medium, the normal. Again, N14 medium was taken. The E. coli was cultured in N14 medium. That means, see, now we have this DNA. Now we have this DNA which has one intermediate type, which is intermediate type. That means one black, the N14 type, and the second one is the red N15 type. We have this hybrid, and the replication will take place in this hybrid and in the normal solution. So what will happen? See, this is the normal nitrogen. This was cultured in N14. Then when it was cultured in N14, again the new strand formed will be N14. This is the new strand formed, N14. So this is what they got. That means ultimately two strands out of this will be formed, two strands out of this will be formed. So what they got, this is one strand with one black. Okay, then another strand is this N50, N14 and N14. Then another strand. Now when we open up this, one is black, one is red. So one black again. So on that, because it is growing in N14 again, so N14. And then another one is again a hybrid. So we got two hybrids, and what we got two hybrids and two normal N14. We got two hybrids and two normal. This is what we have got. Now that means we have two N14. This is completely N14. This is also completely N14. And this is hybrid. That means one red, one black. This is a hybrid, intermediate type. And this is also a hybrid. This is what we got. Now on centrifugation, when this was centrifuged, on centrifugation, in stage 3, what was obtained? In stage 3, what was obtained on centrifugation? Now we have two types. This is one type and this is another type. So in the centrifuge tube, when this was centrifuged, in the centrifuge tube, this is the centrifuge tube. In the centrifuge tube, we will get two bands. Two bands were formed. The first band was the hybrid. Which one will be heavier? Hybrid or the normal one? Hybrid will be heavier. So hybrid took a place here. This is hybrid. This place was taken by the hybrid, and the normal was lighter because normal is N14. The normal took a place somewhere here. So this was a hybrid which has both N15 and N14, whereas the normal had just one, which is N14. So these were the bands which were formed after density gradient centrifugation, and on the basis of this, the Michelson and Stahl, they were able to prove that the replication is DNA in DNA is semi-conservative. One strand is formed new and another strand is formed. One strand is formed new and another strand is old. Is this clear? Michelson and Stahl experiment which I have just explained, is it clear to you all, children? Is it clear to you all, or shall I repeat the experiment again? Or any step which you are not able to understand, that also you can tell me. Yes, beta. Is it clear, or any step needs a repetition? This is a very important experiment of Michelson and Stahl, being asked many a times in the exam. So Vanshika is saying it is clear to her, and others also. Okay, Paras is also saying. I want response from you all. Yes. Any step which you are not able to? Yes, Sukhsahaj. Any step which you are not able to understand in this experiment? Yes, beta. Any step which you are not able to understand in this experiment? Only 3 response I have got. Amit Katoch, is it clear, or any point you want me to repeat? Yes, yes, Parthavi. Any point you want me to repeat in this? Yes, bache? Any point here? The stage 1, stage 2, and stage 3 of the experiment, which was performed by Michelson and Stahl to prove experimentally that DNA replication is semi-conservative. That means one strand is formed new, another strand is the template strand is the old strand on which the new strand is synthesized. So the template strand is the old strand, and on that old strand the new strand is synthesized. This was explained by Michelson and Stahl, and this experiment proves that DNA replication is semi-conservative. Yes, beta? So if this is understood, I'll move on to the next one, next topic, which is replication. So replication, as you know, replication is doubling of DNA. So how does this replication takes place, and what are the enzymes which are used in the process of replication? This we are going to do today, the enzymes, because we have time, at least for the enzymes. So I will erase this for you. Yeah. Now replication. Replication of DNA. DNA shows replication to amplify, to increase in amount, to increase in bulk. It shows the process of replication. Now, replication is of course carried out in the nucleus where the DNA is present. And as a result of replication, the new DNA is synthesized. And of course, the old gets afterwards, it gets disintegrated. The new DNA which is synthesized, this is under the action of many enzymes. Many enzymes are required for the synthesis of for the replication of DNA. The enzymes required for the replication of DNA, the first important enzyme is helicase. Helicase is very important enzyme which is required for the process of replication. After this, there is another enzyme which is topoisomerase. Then we have another enzyme which is SSBP. This is single stranded binding protein. Then we have DNA polymerase. And then we have primer, RNA primer, or primer, which is also known as RNA primer. And after RNA primer, we have ligase. So these are the most important enzymes which are required for the process of this replication. Now helicase. Helicase is the first enzyme which is required for the process of replication, and this brings about the unwinding or unzipping of DNA double strand. The DNA strands, they unwind, they open up and they unzip, and they go apart. Both the strands, they go apart because that is required for the process of replication, and this is done by helicase. When both the strands, they go apart and the unwinding of DNA strand takes place. So this is done by enzyme helicase. Now see, when these are the two strands of DNA, suppose these are the two strands of DNA, when they unwind, when they open up, so they will stretch from here, they will go apart. So when they go towards this side and this side to open up, what happens? This coiling in here, the coiling here, when they are going apart, here the coiling will become tightened. This coiling, it will become more, it will become very, it will tighten over here. So this tightening of coiling, or the coiling which is very near now to each other, the coils which are very near to each other, this will create a tension. Now this tension has to be removed, because a tension is created. What will happen that if the tension is created, then this may alter the process of replication. So for this, the tension needs to be removed, and this tension which is created here, because it is stretched towards this side, so the coiling is present in the form of multiple coils over here a tension is created. So this tension which is created here due to very close coiling of the DNA strands is taken care of by topoisomerase. Topoisomerase makes a cut in the coil, it makes a cut, removes the tension, and then again the sealing takes place, sealing by ligase. So what happens, and as a result, this excessive coiling is removed by topoisomerase. Excessive coiling is removed by topoisomerase. Now once the coiling is removed, now the replication process has to start. So for that, the DNA should remain in this position, otherwise the coiling process will not take place. So that coiling process, that replication process to take place, for the replication process to take place, this DNA strand, it should remain in this position for the time till the replication takes place, and that is brought about by SSBP, single stranded binding protein. This single stranded binding protein keeps the DNA strands in position, that means far apart, so that the process of may properly. So till I have told you about three enzyme side, as both the strands there are in topoisomerase, this topoisomerase it reseals it.